The DNA in the nucleus of a cell contains all the information it requires to carry out life's processes, which are growth, development, maintenance, reproduction, and protection. With all this information, it is no wonder that the length of a cell's DNA is far greater than that of a cell compartment. The DNA in a single human diploid cell contains over 7 billion base pairs divided into 46 chromosomes, which would extend over 2 meters in length if stretched end to end. Yet this massive volume of DNA can be condensed to yield high compact chromosomes through the tight packaging of the DNA. First, the DNA is wound around protein nucleosomes like beads on a string. Then the beads are coiled into a helical structure. And finally, the helical structure is itself supercoiled into a highly packed coiled structure. This highly compact structure allows the DNA to be safely stored in the nucleus and to be divided up during cell division without damaging the DNA. However, such tightly wound DNA does not permit molecules to gain access to individual genes in order to transcribe copies of them as required for protein synthesis. For instance, as a replication fork moves along double-stranded DNA, it creates what has been called the winding problem. Even every 10 base pairs replicated at the fork corresponds to one complete turn about the axis of the parental double helix. Therefore, for a replication fork to move the entire chromosome ahead of the fork would normally have to rotate rapidly. To overcome this problem, cells have specialized proteins to unwind the DNA in specific regions when it needs access to it, while keeping the rest of the DNA molecule tightly wound and out of harm's way. Once the DNA is uncoiled, the DNA double helix itself needs to be unwound to separate it into two individual strands so the information it contains can be accessed. The proteins that carry out this job are collectively known as DNA topoisomerases. DNA topoisomerases are ubiquitous enzymes found in all cell types from viruses to man. These enzymes act to regulate DNA supercoiling by catalyzing the winding and unwinding of DNA strands. DNA topoisomerases can be divided into two groups based on the number of strands that they break. One type of topoisomerase called Topoisomerase I produces a transient single strand break or nick. This break is the phosphodiester backbone allows the two sections of DNA helix on either side of the nick to rotate freely relative to each other using the phosphodiester bond in the strand opposite the nick as a swivel point. Any tension in the DNA helix will drive this rotation in the direction that relieves the tension. As a result, DNA replication can occur with the rotation of only a short length of helix, the part just ahead of the fork. Because the covalent linkage that joins the DNA topoisomerase protein to a DNA phosphate retains the energy of the cleaved phosphodiester bond. Releasing is rapid and does not require additional energy input. In this respect, the rejoining mechanism differs from that catalyzed by the enzyme DNA ligase. So we can summarize this as break one strand of a DNA helix, ATP independent, except for reverse gyrase, and primarily responsible for relaxing positively supercoiled and or negatively supercoiled DNA. while reverse gyrase can introduce positive supercoils into DNA. Mechanism involves rotating the broken strand around the intact strand to relax or unwind the strain on the DNA helix, followed by releasing the ends of the broken strand. And the last one is play an important role in DNA replication and transcription, that is topoisomerase I. There are the subclasses of topoisomerase I. The first one is type 1A enzymes. These are bacterial topoisomerase I, topoisomerase III, and reverse gyrase. The type 1B enzymes, that are eukaryotic and euchiral viral topoisomerase I and archaeal topoisomerase V. A second type of DNA topoisomerase, that is topoisomerase II, forms a covalent linkage of both strands of the DNA helix at the same time, making a transient double-strand break in the helix. These enzymes are activated by sites on chromosomes where two double helices cross over each other. Once a topoisomerase II molecule binds to such a crossing site, the protein uses ATP hydrolysis to perform the following set of reactions efficiently. The first set is it breaks one double helix reversibly to create a DNA gate. The next one is it causes the second nearby double helix to pass through this break. And the third step is it then reseals the break and dissociates from the DNA. In this way, type 2 DNA topoisomerases can efficiently separate two interlocked DNA circles. We also can summarize the class 2 DNA topoisomerases. The first point would be it breaks two strands of a DNA helix. It is ATP dependent, responsible for relaxing DNA. That is, topoisomerase IV is responsible for this incident, as well as introducing either negative, and topoisomerase II is responsible for this event. The next one is mechanism involves passing an intact DNA helix through the gap made by the broken DNA helix, then resealing the strands. It plays an important role in chromosome condensation and in the segregation of daughter chromosomes during cell division. The subclasses of class 2 DNA topoisomerases, the first type 2A enzymes, these are eukaryotic and euchiral viral topoisomerase II, gyrase, the bacterial topoisomerase II, and topoisomerase IV. The type 2B enzymes are archaeal topoisomerase. Thank you for your patience. I followed the book for this class mainly. I have put the link in description below. If you want to buy, this book is available in Amazon. I took information from an article of Jennifer McDowell. You could check out her in Google. If you like the video, give thumbs up, put a comment below regarding the class, and subscribe our channel for upcoming video notifications.